Okay, now we will proceed to the skeletal muscle contraction. Okay, so we already know about the structure of neuromuscular junction. We already know about the structure of sarcomere and also the structure of thick filament and thin filament. So when you know all about that, then you can proceed to the muscle contraction based on the sliding filament hypothesis. So actually this is the one of the mechanism that very important in this chapter lah, for the muscle contraction. Okay. So this is the very summary process. I think I already explained just now. So normally the initial process is that the action potential already reached the synaptic node. So once the action potential already reached the synaptic node, it will light the synapse. When the nerve impulse reach there, calcium ion, you still remember that there are the calcium ion here. Calcium ion will influx into the synaptic node, and then this one will cause the synaptic vesicle synaptic vesicle move to the plasma membrane of the presynaptic neuron, and then release the neurotransmitter, and then the neurotransmitter play the role in muscle contraction is acetylcholine. So the acetylcholine will be released out from the presynaptic neuron and bind to the receptor on the plasma membrane of the muscle tissue, we known as sarcomere. Right. Okay. Once the neurotransmitter already bind toward the binding receptor on the sarcomere, it will trigger the action potential on within the sarcomere. So the action potential will be transferred or propagated along the plasma membrane, go to the T tubule, and then also will down toward the T tubule until it reach the one type of the smooth endoplasmic reticulum that you already learned in Sem 1, be known as sarcoplasmic reticulum SR. Okay, so once the nerve impulse is already reached the sarcoplasmic reticulum, sarcoplasmic reticulum, what is the function? You know that it's store calcium ion, right? This one you still remember? Okay, you remember that sarcoplasmic reticulum containing calcium ion. So when the nerve impulse is reach the sarcoplasmic reticulum, it will trigger the release of the calcium ion from the sarcoplasmic reticulum through the calcium ion channel. Yeah, so the calcium ion will be diffused out from the sarcoplasmic reticulum toward the to the sarcomere. So within all these is a sarcomere already. And then once the calcium ion is already reached the sarcomere, what will do is it will do this process, meaning that the calcium ion will bind toward the troponin complex. When it's bind to troponin, the myosin binding site will be exposed, meaning that the tropomyosin will change its direction, to expose the myosin binding site, so that your myosin head can bind toward the myosin binding site on the actin. So on the actin. So once the myosin head is bind toward the actin molecule, it will move the actin molecule to the center of the sarcomere. Dia akan tolak, macam push, macam tolak lah. You still remember what you can imagine it. Sepatutnya dia dekat sini, dia dekat sini, dia akan tolak dia ke sini. You still remember this actin molecule? You see my hand, this is actin molecule, and then ini myosin head, dia akan tolak ke sini, tolak actin molecule ni kepada center of the sarcomere. So that is why that I say that only actin filament will involve in the movement only. The thick filament, the myosin filament, is maintained at there one. You still remember about the structure of the sarcomere? Only the thin filament will be moved toward the center one. So that is why that we containing the darker A band. So A band is the region of overlapping between the myosin and actin. So this is the place for the overlapping to occur, and then the actin filament will be pushed, or not say push, pull toward the center of the sarcomere, and also for this same of here. So dia yang Niacin head dekat sini akan tolak actin filament to the right hand side, and for this part ia tolak actin filament ke left hand side. It will mush to it. So this is the general basic concept. And then after that, when the mush and also this is the when the myosin head try to move the actin filament toward the center of the try to move toward the actin filament toward the center of sarcomere. This one we call as muscle contraction. So muscle contraction is now is done. Is conducted now. Okay. Once they already complete the muscle contraction, already move to the center, it will go back. The actin filament will go back to the origin location. So how to do this? The myosin head will be released out, not bind to the myosin binding site in the actin already. Can release out, and then the tropomyosin molecule will start to block the myosin binding site on the actin molecule again. And then the calcium ion inside the sarcomere will be pumped out again into the sarcoplasmic reticulum. So this is the recycling process. Unless there are another calcium ion release, then another cycle will be proceed. If no calcium ion, then the muscle is in relaxed condition. So normally this is the basic summary process. So now we will go to the more detailed part. So normally in first, I already mentioned that. So normally in first first step is the releasing of the neurotransmitter bind toward the binding site of the neurotransmitter on the sarcolemma. So normally this process is totally same that you learn in synapse. So you see that electrical impulse reach the synaptic node, and then calcium ion open, and then allow calcium ion go inside the synaptic node, and then the concentration increase, cause the synaptic vesicle fuse with the presynaptic membrane, and then we will release the neurotransmitter. So the neurotransmitter involved is acetylcholine. So all acetylcholine will diffuse across the synaptic cleft and bind the receptor on the sarcolemma, so causing depolarization, so trigger action potential. So the ligand sodium ion channel will be open, so the sodium ion will be diffused into the sarcolemma, so trigger the action potential. So totally same, almost totally same that you learn in synapse. Okay, just that the difference is starting from here. So normally already mentioned, when the action potential is already triggered on the sarcolemma, it will be transmitted along the past sarcolemma until it reach the T tubule, and then it will down to the action potential will move down, speed inward through the T tubule, and then when it reach the sarcoplasmic reticulum, it will cause the calcium ion on the surface of the sarcoplasmic reticulum to open, so that the calcium ion will diffuse through the channel into the cytosol of the muscle fiber. Okay, so once they already enter the calcium ion is already enter, it will go through this 1, 2, 3, 4, 5 process. Okay, so we will see one by one. So first step is that your myosin head will start to bind ATP. You still remember that the structure of myosin head is containing two binding sites. One binding site is actin binding site to bind to the actin filament, and another binding site is ATP. So it will bind to ATP. So this means that myosin head will start to bind ATP. So now when it's starting to bind ATP, it is in low energy configuration. Low energy configuration meaning dia dekat, dia macam tak bergerak lah, dia bukan berdiri punya, dia macam lemah sikit, lenggok punya, macam relax je, tak buat apa-apa lah. Okay, sampai lah ATP relax je lah, tak buat apa-apa lah. Okay, and then at the same time this mentioned, the calcium ion will bind to the troponin complex too. So you will see this diagram, they don't show the troponin complex, but you will still show this, you will still remember this troponin complex. The calcium ion will bind to this troponin complex one. Okay, so once they already bind to troponin complex, the troponin tropomyosin complex will be rearranged. Then will be rearranged again. So will rearrange, the resulting is the tropomyosin will move, it will move and expose the myosin binding site at the globular actin for the establishment of the myosin head. So it will expose to it. So why you see this diagram does not containing tropomyosin already, because tropomyosin is already rearranged. You need to expose the binding site of the myosin. So you see that all the actin filament does not containing the tropomyosin already. Or now the myosin head now can start to bind to the actin filament again. So this is the first step. So next, second step. Okay, once in second step, the myosin head will start to hydrolyze ATP to form ADP and phosphate group. So it will start to using ATP as energy already. So when it is starting to use ATP, so that it is now in high energy configuration. You see this diagram, dia bergerak, dia tegak. Tadi dia lengah-lengah sikit kan, sekarang dia dah menjadi fokus lah. Tadi in focus situation, dia nak bergerak, starting to bind to the myosin binding site on the actin filament already. So the myosin will bind to the binding site of myosin at the globular actin. So it will bind to the actin filament lah, and then this one will form the cross bridge. So it will, once it bind to the binding site of the myosin on the actin, it will form the cross bridge. Dia akan satu jambatan between them. So like this, you will see that it will form the cross bridge, so that this cross bridge is formed within the thin filament lah. It will form the cross bridge. After that, this myosin head will start to move the actin filament toward the center of the sarcomere. So the myosin will start to release the ADP and phosphate group to power suit that slide the thin filament toward the center of the sarcomere. So you still remember, it will myosin head hydrolyze ATP to form ADP and phosphate group, but for now ADP still bind toward the myosin head. But when the contraction is starting to occur, the ADP and phosphate group will be released out from the myosin head, and this one will cause the myosin head slide the thin filament toward the center of sarcomere. So that is why that we known as sliding filament hypothesis. Slide is the key terms that you need to use. So just now I say push and pull, right? You cannot use push and pull. You need to use slide the actin filament toward the center of sarcomere. And then on the other hand, myosin head will now return to low energy state. So once the myosin head is already doing the sliding process, then the myosin head will become dah lemah lah, tak ada energy lah, dah buat slider, tak ada energy lah, so in low energy state lah, tak ada energy lah, tak boleh makan lah, tak ada nak makanan lah. So apa makanan dia? ATP lah. So later in low energy state, so another new ATP will bind to myosin head again. So the cross bridge now will be disrupted. So once they already finish the sliding due to the low energy configuration, ATP bind to the myosin head, and then it will become, the cross bridge will be disrupted, and then the myosin head will be released from the actin filament. Okay, so when the presynaptic neurons stop to release acetylcholine, so this one, when the process is already stop release acetylcholine, so no nerve impulse lah. So when no nerve impulse within this T tubule with no acetylcholine already, so no action potential, to trigger the calcium ion, so that if no action potential, the calcium ion inside the cytosol of the muscle fiber will be pumped back into the sarcoplasmic reticulum through the carrier protein by active transport. Okay, and then so when the calcium ion inside the cytosol will decrease, causing the regulatory protein bound to the thin filament shift back to starting position. So the regulatory protein is troponin. Troponin will bound to thin filament again, and then the tropomyosin will block the myosin binding site again. So it will return back to the origin position that you see like this. Like this. This is the origin location. Okay, so now the muscle now is relaxed, and then this process will be repeated again when the neurotransmitter is released, what you call as acetylcholine. It released from the presynaptic neuron, and then the sarcoplasmic reticulum receive the calcium ion again. So the whole process will be continued again. So it will repeat the process once the calcium ion is released from the sarcoplasmic reticulum and bind to the troponin molecule. If no, then the muscle is in the relaxed position like this. And then this lower diagram is the contraction. So when you see this, what are the difference is? Main difference is that the actin filament now is moved toward the center, right? At the origin location, you see this M line, at the M line, middle line, and also H zone. H zone is totally containing only thick filament during the relax, and especially H zone. But for now, active filament is slide to the center of the sarcomere. So who slide the actin filament to the center? Myosin head lah. So you see this, all the myosin head now is already form the cross bridge between the myosin and also actin filament. You see this, all this, so that it will move toward the center. So you see that the length of the sarcomere now is more shorter. Though this is very important. Normally in structural question it will make you to differentiate these two. So you need to know lah. Okay, next